In today's video for lesson 3.3, we are going to be focusing on regulation of the cell cycle. So if you remember, cell cycle includes 3 phases. We have interphase, mitosis, and cytokinesis. So what we're talking about today is how do cells know when to divide and when not to? And then what happens if this regulation system we have fails? So when we're talking about the regulation of the cell cycle, we need to know that the cell cycle is controlled by a chemical control system that starts and stops the events in the cell cycle. So weird to think about, but everything that happens in your body is the result of some sort of chemical reaction occurring. And in addition to these chemicals, we are also going to have proteins that play a key role. And if we remember, proteins, we said, have so many functions and they are running our body. So regulation of the cell cycle happens in a couple different ways. So first, we can have external regulation, and these are signals that come from the outside of the cell. So you're very familiar with these. These are things like hormones, nutrients, whatever it may be, they're coming from outside of the cell. And the other option is internal, and these are signals that come from within the cell's nucleus. So it's telling itself, so this example would be DNA. As our cell goes through the cell cycle, it's going to go through a series of checkpoints. And a checkpoint is a critical point where stop and go signals help regulate the cycle. So we have our cell cycle here unlabeled. And if we go through and label these, we always start with interphase. And of interphase, that's in blue, we're going to go with G1. Then we will have our first resting phase. From G1, we will move to S. From S, we move to G2. So we have one at the beginning of interphase, making sure we're ready for our DNA replication in S and synthesis. And then we have one at the end to make sure that we did this correctly. And then after we go through our interphase, then we have our mitosis and cytokinesis, because remember, we lump those together. So we have G1, S, G2, and mitosis. But if we look, we're spending most of our time in this blue part of interphase here. So most of cell division is spent in the off position because there's no stimulus present. So we spend most of our cells not dividing. And as we're talking about cell regulation, we also need to talk about what we're going to do if something goes wrong. And one of our options is apoptosis. And if we remember, apoptosis is programmed cell death. So we're going to look at examples of when our body does apoptosis. So it is an internal or external signals that can activate genes that produce these self-destructive enzymes. So apoptosis can be controlled internally from the DNA or externally coming from hormones or just some other molecule that's going to produce these enzymes that are going to break down the cell. And so our example that goes with this is the formation of webbed fingers and toes during development. So something's going wrong here. There's some reason your body decided to go through apoptosis in the location of your toes, and that is going to end up with you having webbed toes. So anybody that has webbed fingers or toes, they're born with it because apoptosis happened during development. So unfortunately, one of the things we have to talk about when talking about regulation of the cell cycle is cancer. And cancer we define as uncontrolled cell division, and this happens when the cell cycle regulation breaks down. So we no longer have control over what is happening within the cell cycle. So if you remember, we spend most of our time in interphase normally and not mitosis, but when our cells are cancerous, that's not happening, and so we're going to spend more and more time dividing, creating more and more cells. Which means that our cancer cells divide much more often than our healthy cells do, and this is going to cause tumors to form. And we define a tumor as a clump of cells that divide uncontrollably. So they're going to be more often than our normal cells. And we will talk about the difference between tumor and cancer. We're going to talk about some different types of tumors and to specify when we should be scared of this word and when you don't necessarily need to be. So our first type of tumors are benign, and these are abnormal cells that typically remain clustered together. So these are typically harmless and can be easily removed. So an interesting example of a benign tumor are moles. They are abnormal growths, but they stay together in that same location. They're not spreading and typically harmless and easily removed. So having the word tumor come up in a medical discussion doesn't always have to mean that it's a bad thing. Our second type of tumor are called malignant tumors, and these are cancer cells that have broken away from their tumor and are moving to other parts of the body, which means that they are causing more tumors. So if we are using this word malignant, or that means that it is cancerous rather than just being this harmless type of tumor. And another important word to understand when talking about cancer and tumors is metastasize. And this is the spreading of disease from one organ to another. So this is a major characteristic of cancer, the ability of it to spread from one organ to the next, which is a defining factor of our malignant tumors comparatively to our benign tumors. So we often hear of this word cancer and we're scared of it, but let's talk about some of the things that can cause cancer so we can be better prepared to make choices throughout our life to prevent this from happening. So the first factor that we are going to talk about are called biological factors, and these are things like age, inherited genetic mutations, skin type, etc. So anything that is caused by your genetics is going to be a biological factor. These ones, for the most part, you can't really do much about, and it's just kind of luck of the draw whether you got good genes or not. Our second factor going into causes of cancer are lifestyle choices. These we do have control over, so things like diet, physical activity, exposure to UV radiation. That's a big one, something we can entirely prevent. So UV radiation, if we don't know, that's sunlight, and so anytime you are exposed to sunlight and you have a change in skin color, so whether you be burn or you tan, any change to skin color is a mutation to your DNA, which is leading to cancer. So it is not worth having a tan that could potentially cause cancer. And then we have viruses and other infections that can lead to cancer. So an example of this is HPV, or the human papillomavirus, which can lead to cervical cancer. And then lastly, our last factor is exposure to carcinogens. And this big long word, carcinogens, means cancer-causing agents. And these are chemicals that cause cancer by mutating the DNA. So just like UV radiation, carcinogens is the category for this. So it can be things like tobacco smoke, asbestos, sunlight. Any chemical that causes cancer is a carcinogen. And so I've included pictures here. We have sunlight for UV radiation, cigarette smoke, and there are many, many others. And so we want to limit our exposure to these carcinogens to help limit our possibilities of ending up with cancer.